Hey everyone, welcome back for another deep dive. And today we're going to be looking into something really fascinating, a drug discovery success story.
Yeah.
Um, Specifically, the development of an HIV protease inhibitor.
Right.
And this is it's like a detective story,
Yeah.
really figuring out how to stop this tiny but formidable foe.
Yeah, it's a great example of how scientists take an abstract concept, in this case, understanding a virus,
Right.
and translate that knowledge into an actual tangible solution, like a drug that can actually fight the disease.
Yeah, so not just theory, we're going deep into like the nitty-gritty of how a drug actually goes from concept to reality.
Absolutely. And to make it even more intriguing, we'll compare this successful drug to one that actually failed during development.
Oh, interesting.
Even though it showed early promise, so it'll be like the side-by-side comparison of what separates a winner from a dud.
Okay, I'm hooked already. We've got
Yeah.
excerpts from research papers,
Yeah.
medicinal chemistry textbooks, even a peek into preclinical drug development handbooks.
Wow.
So where do we even begin with a story like this?
Well, let's set the stage. Imagine HIV as like a microscopic machine, relentlessly trying to make copies of itself.
Okay.
One crucial tool in its arsenal is something called protease. It's an enzyme that acts like a pair of molecular scissors.
Scissors.
Oh.
Okay, that's a good visual. What are they cutting?
So HIV protease, it snips long protein chains into smaller functional pieces, and these pieces are essential for the virus to mature and spread.
So it needs to do that in order to replicate.
Exactly. And if you block that, it's like disrupting a communication pathway, preventing the virus from sending out the signals it needs to replicate.
Got it. So if you can stop those scissors, you stop the virus from replicating. That's where protease inhibitors come in.
Precisely. Scientists recognized early on disrupting this process would be key to fighting HIV.
Yeah.
It's like, choosing the right battle to fight, going after a critical vulnerability in the enemy's defenses.
Right. Target validation.
Exactly.
That makes
Yeah.
sense. But actually designing a molecule that can effectively block HIV protease, that's where the real challenge lies.
Oh, absolutely. It's not like scientists can just, grab any molecule off the shelf and hope for the best.
Right.
This is where drug design gets really interesting. It's a blend of creativity and, meticulous scientific understanding.
Right.
Scientists had to unravel the 3D structure of HIV protease, map out its shape, how it interacts with the proteins it cuts.
So it's like building a lock and key, where the key is the drug molecule and the lock is that active site of the protease.
That's a great analogy, but here's where things get even more intricate. Early attempts at designing HIV protease inhibitors focused on molecules that mimicked the natural protein pieces that the protease usually cuts.
Okay, so if it looks like the target, it should fit into the active site, right? But I have a feeling there's a but coming.
You read my mind. The problem was these early inhibitors, being so peptide-like, were quickly chewed up by the body's own enzymes.
Oh.
It was like sending a soldier into battle wearing a uniform made of paper. It wouldn't last very long.
So their own weapon was getting destroyed before it could even reach the enemy.
Exactly. It was a major setback. Scientists realized they needed a more durable weapon, something that could withstand the body's defenses and still reach its target.
Right.
This led to a shift toward designing non-peptide, like inhibitors molecules that could bind to HIV protease, but wouldn't be as easily degraded.
So it's not just about finding a molecule that binds it has to have the right properties to survive in the body as well.
Exactly.
That's where this whole idea of lead optimization comes in.
Right. Think of it like refining a recipe.
Okay.
You start with a basic idea, but then you tweak the ingredients, the proportions, the cooking time until you achieve the perfect flavor and texture. In drug development, those ingredients are the molecule's properties.
Okay, so what kind of properties are we talking about? What makes a good drug molecule recipe?
Well, one key property is something called metabolic stability.
Okay.
Basically, how quickly the drug is broken down by the body. Remember those early peptide-like inhibitors we talked about?
Yeah.
They had poor metabolic stability because they were rapidly degraded by enzymes.
Right, like the paper uniform just disintegrating in the rain.
Exactly. So scientists had to find ways to make the inhibitor more resistant to those enzymes.
But wouldn't that focus on metabolic stability potentially overlook other important properties of the drug?
That's a great question, and it highlights the delicate balancing act in drug development. You have to consider a whole range of properties, not just metabolic stability.
So what else is on the checklist?
Well, you also need to think about how well the drug is absorbed into the bloodstream, how it distributes throughout the body, and whether it reaches the target tissues in sufficient concentrations.
It sounds incredibly complex, like trying to solve this multi-dimensional puzzle where each piece has to fit perfectly to create a successful drug.
That's a great way to put it, and To complicate matters further, scientists also have to be mindful of potential toxicity.
Right.
Even if a drug is effective, it's no good if it causes harmful side effects.
So it's about finding that sweet spot, a drug that's effective against the virus, able to survive in the body, and safe for the patient.
Exactly. And finding that sweet spot often involves a lot of trial and error, a lot of testing and refining.
Hmm.
To see how this plays out in the real world, let's take a closer look at an HIV protease inhibitor that successfully navigated this complex development process.
Okay, let's meet our star player. What's the name of this successful drug?
This particular protease inhibitor is called sequinavir.
Sequinavir. Okay.
And its journey began, as we discussed, with scientists identifying HIV protease as a key target. They knew they had to find a way to disrupt this molecular machine if they wanted to effectively fight the virus.
And sequinavir was the molecule that ultimately emerged victorious. It must have been a fierce competition.
You could say that sequinavir's journey is a testament to the power of scientific ingenuity and perseverance, but as we'll see, it wasn't a straight path to success.
Okay.
There were unexpected twists and turns, moments of doubt, and even a few near misses along the way.
Ooh, suspense already. It's getting good. Take us back to the beginning. How did sequinavir first come onto the scene?
Well, the story of sequinavir really picks up steam in the lab, where scientists were hard at work, designing and testing potential protease inhibitors.
And I'm guessing this wasn't a straightforward process. What were some of the roadblocks they encountered?
As we talked about earlier, those early attempts at designing peptide-like inhibitors hit a major snag. They were just too unstable in the body, quickly broken down by enzymes before they could reach their target.
Right, we talked about that. So how did they overcome this hurdle? Did they have to completely rethink their approach to drug design?
They did have to get creative. They couldn't just abandon the idea of targeting HIV protease.
Right.
So they needed a way to design inhibitors that were less peptide-like and therefore less susceptible to degradation.
Okay, so that's a tough challenge. It's like trying to create a new language that the body's enzymes don't understand.
That's a great analogy. Scientists had to essentially trick the body design a molecule that could still bind to HIV protease, but wouldn't be recognized as a target for those pesky enzymes.
So how did they go about doing that? Did they have some sort of molecular disguise kit?
Well, they didn't have a disguise kit, but they did have a powerful tool at their disposal: medicinal chemistry.
Yeah.
This field combines organic chemistry with an understanding of biological systems to create new molecules with specific properties.
So they were like molecular architects designing and building these inhibitors from the ground up, carefully choosing each atom and bond to achieve the desired outcome.
Precisely. They had to consider not only how the molecule would bind to HIV protease, but also how it would interact with the body's enzymes, how it would be absorbed and distributed, and a whole host of other factors.
It sounds incredibly intricate, like a delicate balancing act between so many different variables.
It certainly was, and as they tweaked and refined their designs, they were constantly testing and evaluating their progress. This wasn't a one-and-done process. They were constantly generating data, analyzing the results, and using that information to guide their next steps.
So they weren't afraid to go back to the drawing board if something wasn't working.
Not at all. In fact, that's a hallmark of successful drug discovery, the willingness to embrace failure as a learning opportunity. They knew that every experiment, even the ones that didn't yield the desired results, was bringing them closer to their goal.
It's like they were detectives following each clue, each piece of data until they cracked the case.
I like that analogy. And remember, we're talking about a time before the advent of high-throughput screening and other technologies that have since revolutionized drug discovery.
Oh, wow. So they were doing all of this manually testing one molecule at a time.
To a large extent, yes. It was a painstaking process that required a lot of patience and persistence.
Wow, that's incredible. It really underscores the dedication and ingenuity of those early pioneers in HIV drug discovery.
Absolutely. And as they meticulously pieced together their molecular puzzle, they started to see some promising leads emerge. One of these leads would eventually become sequinavir.
Okay, so we've got our hero sequinavir stepping into the spotlight. What were its vital stats? What made it stand out from the crowd of other potential inhibitors?
Well, early tests showed that sequinavir had a strong affinity for HIV protease, meaning it bound tightly to its target. That's a crucial first step, of course.
Right, it has to be able to grab onto the protease and hold on tight to prevent it from doing its job. But binding affinity is only part of the story, right? What else was going for sequinavir?
Remember those metabolic stability studies we discussed? Well, sequinavir performed admirably in those tests as well.
Okay.
It wasn't as easily broken down by enzymes, meaning it could potentially last longer in the body and have a greater chance of reaching its target.
So it was shaping up to be a strong contender, good binding affinity, decent metabolic stability. What happened next?
Well, as promising as sequinavir looked in the lab, the real test was how it would behave in living organisms.
Right, because what happens in a test tube doesn't always translate perfectly to what happens in a complex biological system.
Exactly. So the next stage involved testing sequinavir in animal models to see how it was absorbed, distributed, metabolized, and eliminated. Basically, they wanted to get a feel for its pharmacokinetic profile.
Okay, so it was time to see if sequinavir could perform on the real battlefield, not just in the controlled environment of the lab. What did those animal studies reveal?
Well, the results were encouraging, but they also revealed a potential challenge. While sequinavir was generally well tolerated, its bioavailability, the amount of drug that actually reaches the bloodstream, was somewhat limited.
So even though it showed promise in the lab and in animal models, there was a chance that it wouldn't be absorbed well enough in humans to be effective. That's a tricky situation.
It was, and it's a common problem in drug development sometimes. A drug that looks great on paper just doesn't quite make it over that final hurdle. But the researchers weren't ready to give up on sequinavir just yet. They knew they had a potential game changer on their hands, so they set out to find a way to improve its bioavailability.
Okay, so how did they boost its chances? Was there some secret ingredient they could add to the mix?
Well, it wasn't a secret ingredient per se, but it did involve a clever strategy, a combination therapy.
Intriguing. Tell me more.
They discovered that by combining sequinavir with another drug called ritonavir, they could significantly enhance its bioavailability. Ritonavir inhibits an enzyme that breaks down sequinavir, essentially allowing it to stay in the bloodstream longer and reach higher concentrations.
Wow, that's a brilliant workaround. It's like giving sequinavir a bodyguard to protect it from those degrading enzymes.
That's a great way to think about it. This combination therapy proved to be a game changer, paving the way for sequinavir's eventual approval as an HIV treatment.
This is a truly inspiring story of scientific ingenuity and perseverance. But you mentioned earlier that we'd also be looking at an example of an HIV protease inhibitor that didn't make it. Can you tell us about that?
Certainly, while sequinavir stands as a testament to the power of rational drug design and lead optimization, there are other HIV protease inhibitors that faced a different fate. One such example is a compound that never even made it to clinical trials.
Oh, wow. It failed even before reaching human testing. What happened?
Well, during preclinical testing, researchers discovered that this particular protease inhibitor exhibited significant toxicity to heart cells.
So it was attacking the very cells it was supposed to be protecting. That's a serious problem.
Yes, and unfortunately, this toxicity was severe enough to halt further development of the drug, despite its potent anti-HIV activity. The risk of cardiac side effects was deemed too high to justify moving forward with human trials.
This is a stark reminder that drug development is a delicate balancing act. You need a drug that's effective against the disease, but also safe for the patient.
Exactly, and sometimes, despite the best efforts, a drug candidate reveals unforeseen toxicities that preclude its further development. Comparing sequinavir's success with the failure of this other protease inhibitor highlights the critical factors that can make or break a drug.
I'm curious, what were those key differentiating factors? What set sequinavir apart from the one that failed?
Well, in sequinavir's case, meticulous lead optimization played a crucial role. Scientists systematically assessed its properties, including metabolic stability, absorption distribution, and potential for toxicity. They fine-tuned its structure to achieve a balance between efficacy and safety.
So they were constantly evaluating and adjusting their strategy based on the data they were gathering.
Precisely, they were adapting their approach in real time, responding to the challenges as they arose. This iterative process of testing and refining is at the heart of successful drug development.
And in the case of the protease inhibitor that failed, did they miss something during the lead optimization stage? Did they not adequately test for cardiac toxicity?
It's not always a case of missing something. Sometimes toxicities emerge unexpectedly, even with rigorous testing. Remember, preclinical studies are often conducted in animals, which might not perfectly predict how a drug will behave in humans.
So even with all the advances in drug design and testing, there's still an element of uncertainty.
Absolutely. Drug development is a complex and challenging endeavor, and there's always the possibility of unforeseen hurdles. But by learning from both successes and failures, scientists can continuously improve their strategies and increase the chances of bringing safe and effective drugs to patients.
This has been incredibly insightful. We've really gotten a glimpse into the intricate world of drug development with all its triumphs and setbacks.
Indeed, and I think it's important to emphasize that even seemingly small changes to a drug's structure can have a huge impact on its success or failure. It's a testament to the power of chemistry and the dedication of scientists who work tirelessly to develop life-saving medications.
Well, we've covered a lot of ground in this first part of our deep dive. We've explored the challenges of designing HIV protease inhibitors, the intricate process of lead optimization, and the delicate balancing act between efficacy and safety. But there's so much more to unpack.
Indeed, we've only just begun to scratch the surface of this fascinating story.
We'll be back soon to delve even deeper into the world of drug discovery success.
You know what's fascinating about saquinavir's success? It really highlights the importance of adaptability in drug discovery.
Adaptability. What do you mean by that?
Well, scientists encountered several unexpected challenges along the way, like that bioavailability issue we discussed.
Right.
They could have easily given up when they realized saquinavir wasn't being absorbed well enough, but instead they found a creative solution through combination therapy.
Yeah, that's a great point. It wasn't a straight line from idea to finished drug.
Right.
It sounds like they had to constantly adjust their approach based on the data they were gathering.
Exactly. And that's a crucial lesson for anyone involved in drug discovery.
Mhm.
You know, the willingness to pivot, to try new things, to not be afraid to deviate from the original plan when the data suggests a different course of action.
So being a good drug hunter is as much about being flexible and resourceful as it is about scientific expertise.
Absolutely. It's a blend of scientific rigor and creative problem solving. Sometimes the most groundbreaking discoveries come from those unexpected detours, those moments where you have to think outside the box.
That's really inspiring. It reminds me of a quote I once heard. The greatest discovery of all time is that a person can change his future by merely changing his attitude.
I like that it applies perfectly to the world of drug discovery. And speaking of changing attitudes, there's another aspect of saquinavir's development that I think is worth highlighting, the shift in thinking about drug targets.
Oh, interesting. How did the understanding of drug targets evolve during this time?
Well, when scientists first started working on HIV treatments, they focused primarily on targeting viral enzymes like reverse transcriptase and protease.
Okay.
These enzymes are essential for the virus's life cycle, so blocking them seemed like the most logical approach.
Makes sense to go after the key players, disrupt the enemy's operations.
Mhm.
But you said there was a shift in thinking. What changed?
As scientists learned more about HIV, they realized that the virus is a master of disguise and adaptation. It can mutate, rapidly developing resistance to drugs that target a single enzyme.
Ah, so it's like a moving target constantly changing its shape to evade those molecular bullets that makes things much trickier.
Exactly. So scientists started exploring new approaches, looking beyond single enzyme targets.
Hmm.
They began to consider targeting host factors, proteins in the human body that HIV relies on for its survival.
So instead of attacking the virus directly, they started looking for ways to weaken its support system, cut off its supply lines.
Precisely. It's a more indirect approach, but one that holds a lot of promise for overcoming drug resistance.
Okay.
The idea is to target cellular pathways that are crucial for HIV, but not essential for human cell survival.
That's fascinating. It's like cutting off the enemy's food supply without harming your own troops. But I imagine targeting host factors comes with its own set of challenges, right?
You're right, it's a more delicate balancing act. You have to ensure that inhibiting the host factor doesn't disrupt normal cellular processes and cause harmful side effects.
So it's like walking a tightrope between effectiveness and safety. But if scientists can pull it off, it could be a game changer in the fight against HIV.
Absolutely. It's one of the most exciting frontiers in HIV drug development today, and it speaks to the constant evolution of scientific thinking, the willingness to challenge old assumptions and explore new avenues.
This is really incredible. It's like we're witnessing a scientific arms race with scientists constantly developing new strategies to outmaneuver this elusive virus.
That's a great analogy, and just like in any arms race, the development of new weapons, or in this case, new drugs, often leads to countermeasures from the enemy.
Meaning HIV will likely find ways to develop resistance to these new host factor inhibitors, just like it did with earlier drugs.
It's certainly a possibility, but scientists are already thinking ahead, trying to anticipate those moves and develop even more sophisticated strategies to stay one step ahead.
So it's a never-ending battle, a constant back and forth between human ingenuity and viral evolution.
You could say that, but that's what makes this field so exciting. There's always a new challenge to overcome, a new mystery to unravel, and the stakes couldn't be higher.
Well said. And I have to say, this conversation has given me a whole new appreciation for the incredible complexity and ingenuity involved in drug discovery.
It's a truly remarkable process, and as we've seen with Queen story, it's one that's driven by passion, perseverance, and the unwavering belief that we can find solutions to even the most formidable challenges.
This deep dive has been incredibly illuminating. We've explored the triumphs and setbacks of HIV drug discovery, witnessed the evolution of scientific thinking, and glimpsed into the future of this ever-evolving field.
It's been a pleasure sharing these insights with you, and I hope it's inspired our listeners to learn more about this fascinating world where science and human ingenuity converge to create life-saving solutions.
Absolutely. But before we wrap up this episode, there's one more area I'd like to touch upon: the role of collaboration in drug discovery.
That's a great topic. Collaboration is absolutely essential in this field. No single scientist, no single lab, no single company can do it alone. It requires a collective effort, a pooling of knowledge and resources from across disciplines and organizations.
So it's like a global scientific community coming together to tackle these complex challenges.
Precisely. And we see this collaborative spirit at play in so many aspects of drug discovery, from the initial stages of target identification to the clinical trials that ultimately determine a drug's fate.
Can you give us some specific examples of how this collaboration plays out in the real world?
Certainly, one example is the sharing of data and research findings. Scientists often publish their work in scientific journals, making their discoveries accessible to others in the field.
So it's like a giant open source project where
Hmm.
everyone contributes their piece of the puzzle and together they build a more complete picture.
That's a great way to think about it. And beyond publications, there are also numerous conferences and workshops where scientists from all over the world gather to share their latest findings, discuss challenges, and brainstorm new ideas.
It sounds like a vibrant and dynamic community constantly pushing the boundaries of knowledge and innovation.
It truly is. And these interactions, these exchanges of ideas and perspectives are often the spark that ignites new discoveries.
So it's not just about the hard science, it's also about the human connections, the relationships that are forged through these collaborations.
Absolutely. Science is a human endeavor driven by curiosity, passion, and the desire to make a difference in the world. And those qualities are amplified when people come together, share their knowledge, and work towards a common goal.
This has been such an enlightening conversation. It's clear that collaboration is the lifeblood of drug discovery, fueling innovation and accelerating progress.
It's been a pleasure exploring this topic with you, and I think it's important to remember that behind every successful drug, there's a team of dedicated scientists, clinicians, and regulatory experts all working together to improve human health.
Well said. And as we wrap up this part of our deep dive, I'm struck by the incredible journey we've taken. We've explored the intricacies of HIV protease, the challenges of drug design, the importance of adaptability and collaboration, and the promise of new approaches like host factor targeting.
It's been quite a ride, and we've only just begun to scratch the surface of this fascinating field.
I'm eager to see what other discoveries and breakthroughs await us in the future.
I share your enthusiasm. The world of drug discovery is constantly evolving, and I have no doubt that there are many more exciting chapters yet to be written.
Well, we'll be back soon to delve even deeper into this fascinating world. As we were talking about all these complexities of drug discovery, I keep thinking about just the sheer amount of testing and analysis that goes into developing a single drug.
Yeah, it's really mind-boggling when you consider the number of molecules that are screened and evaluated before even a single drug candidate emerges.
It's like searching for a needle in a haystack, but on a molecular scale.
Right.
And even when you find that needle, there's no guarantee it will work as intended.
Absolutely. It's a high-stakes game with a lot of potential pitfalls along the way.
Right.
But thankfully, the tools and techniques for drug discovery are constantly evolving, becoming more sophisticated and efficient.
Speaking of tools, you mentioned earlier that technologies like high-throughput screening have revolutionized drug discovery. Can you tell us a bit more about that?
Sure, high-throughput screening, or HTS, it allows scientists to rapidly test thousands, even millions of compounds against a specific target like HIV protease.
Wow, so it's like having a robot army of scientists working around the clock, conducting experiments at an incredible pace.
That's a great analogy. HTS has dramatically accelerated the early stages of drug discovery, helping scientists identify promising leads much faster than traditional methods.
That's amazing, but I imagine it still takes a lot of work to go from a promising lead to an actual drug that can be used to treat patients.
Oh, absolutely. HTS is just the first step in a long and complex journey. Once a potential drug candidate is identified, it undergoes a very rigorous series of tests to evaluate its safety and effectiveness.
So it's like putting the drug candidate through boot camp, testing its limits and making sure it's up for the challenge.
That's a great way to put it. These tests include preclinical studies in animals followed by clinical trials in humans, and at each stage there's a possibility that the drug candidate will fail to meet the required standards.
So even with all these advances in technology and screening methods, there's still a high degree of uncertainty in drug development.
It's a humbling reminder that we're dealing with very complex biological systems, and there's always the potential for unexpected outcomes. But despite the challenges, the progress that's been made in HIV drug discovery over the past few decades is truly remarkable.
It really is. It's incredible. It's a testament to the dedication and ingenuity and collaborative spirit of scientists worldwide.
Yeah.
And as we wrap up this deep dive, I'm left with a sense of awe and wonder at this intricate world of drug discovery.
It's been a pleasure exploring this world with you, and I think the key takeaway is that drug discovery is a very dynamic and ever-evolving field.
Yeah.
It's driven by this relentless pursuit of knowledge and a commitment to improving human well-being.
Well said. And I want to thank our listeners for joining us on this fascinating exploration. We hope you've gained a deeper appreciation for the complexities and wonders of drug discovery.
And as always, we encourage you to continue learning and exploring this ever-fascinating world. Who knows what groundbreaking discoveries await us just around the corner?
That's right. So keep that curiosity alive, and we'll catch you on the next deep dive. Until then, stay curious and keep learning.